Hi everyone, I'm Ummey. I'm a PhD student in the School of Medicine. One of the aims of my PhD project is developing a quick, cost-effective genetic screen for Amelogenesis imperfecta, or AI for short, a disease leading to defects of the teeth. Before I go into more details, I'd like to thank my supervisors, Dr. Alan Mile, Professor Chris Engelhardt, and Dr. Christopher Watson, as well as my co-workers for their support in this work. We are Leeds Dental Genetics Group, which is a collaboration between Leeds Dental Institute and School of Medicine. Our work focuses on understanding the processes and pathways of tooth development and how they can go wrong in tooth diseases. I should also mention here the contributions made by previous group members, James, Dave, Walid, George and Claire on AI research, which has led to the Leeds group to discovering nearly half of all the AI-causing genes and establishing the first comprehensive AI mutation database here in Leeds. Finally, a big thanks to my funders, Leeds Doctoral Scholarship and Wellcome Trust. We can all agree that our smile reflects our personality and feelings. Nothing is more attractive to others than our smile, and a nice smile is a good positive way to introduce ourselves. The children in the picture look very happy and are enjoying themselves, clearly reflected in their lovely smiles. My research is dedicated to ensuring healthy smiles for all by, as I said, focusing on this group of dental conditions, collectively known as Amelogenesis imperfecta or AI, which cause enamel failure. So, what is amelogenesis? Amelogenesis is the process by which enamel is laid down on the surface of the teeth. Enamel formation results from the secretion of an extracellular matrix by secretory cells called ameloblasts and its eventual mineralization. Enamel is the hardest and most mineralized tissue in our body. Amelogenesis stops at the point of tooth eruption, and there is no production of secondary or regenerative enamel from that moment on. It has to last a lifetime. What we know from the earliest history is teeth were not individual structures like what we see now. They were rather tough, bumpy plates that ancient fish used, like sandpaper, to crush and shred their food. The genes involved in tooth development in mammals are similar to those involved in the development of fish scales. And also a study of a tooth plate from a fossil of the extinct fish Romandina stellina showed that the teeth and scales were made up of same tissues, also found in mammal teeth, supporting the theory that teeth evolved as a modification of scales. So what is an AI? AI is a group of rare inherited dental conditions causing enamel failure. It is caused by defects in the genes that are involved in amelogenesis, that is the making of enamel. Affecting around 1 in every 2,000 people, AI appears in infancy upon eruption of the first teeth and worsens with further tooth eruptions through childhood, adolescence, and early adulthood. It can affect both the quality and the quantity of enamel. AI mostly occurs in isolation in the absence of other health problems, referred to as non-syndromic AI, but it can be associated with other significant health problems and would then be called syndromic AI. There are currently 20 non-syndromic and 13 syndromic AI genes discovered so far, and mutations in any of those genes can lead to AI. So, what happens when someone has AI? Depending on the genes involved, AI can be clinically many different types. The teeth are discolored, as you can see in the picture. AI also causes the enamel to be very weak, so it breaks down quickly, causing pain, infection, and early tooth loss. It can also cause misalignment of teeth. There are also well-documented adverse psychosocial impacts of AI on patients with high levels of distress, social avoidance, discomfort, isolation, and emotional problems. As I'm sure you can imagine, the impact of AI on smiles like this is huge, and inevitably, that will have a big effect on the social confidence of people affected by AI. And you might be thinking that you can just replace them with false teeth, but in reality, there's no sound foundation for the dentures to fit onto, so dentists find AI very hard to treat. Although we have identified many of the genes involved in AI, and so understand the disease much better, treating AI is still very complicated. It helps though to identify the correct AI type in each case, which is only possible through DNA sequencing. Next generation DNA sequencing, or NGS, is a revolutionary technology for determining the order of nucleotides in the DNA. NGS is groundbreaking because it makes it possible to sequence millions of pieces of DNA in one go instead of sequencing them one after another. However, the human genome is enormous, and reading all 3 billion nucleotides or all 25,000 genes routinely for many patients is still something very time consuming and expensive. There are also ethical issues associated with genome-wide screening because it can uncover other genetic problems that were not the target of the original investigation. So there was a need to develop a quick, cheap method that would only target the regions of interest and could act as a first point of screening for AI patients. Single molecule molecular inversion probes, or SMAPs, is an adaptation of the NGS method that allows selective screening of lots of regions of the DNA in one single reaction. It involves designing oligonucleotide probes targeting many genomic regions of interest. Each of the probes consists of a common DNA backbone flanked by target-specific sequences located at both ends called extension and ligation arms. These arms aid probe hybridization at the target sequence, and with the help of a DNA polymerase, the gap gets filled between the arms. Thousands of probes designed in this way can be pulled together in a single tube for hybridization with the genomic DNA and can be sequenced afterwards. SMMIP is highly multiplexable, meaning that it allows us not only to selectively screen a lot of regions of the DNA that we are interested in, but also to do that in lots of different people simultaneously. We have developed a reagent consisting of around 700 of these SMMIP probes targeting the 20 known AI-causing genes, and we tested these reagents successfully on 20 patients, finding several new and known mutations in them. So what is next? We now want to scale it up and screen over 300 new patients already recruited from multiple centers across the country. I would probably have done most of that by now if it wasn't for the lockdown. A quick diagnosis of known AI genes will help us to know exactly what type of AI someone has, what are the risks there to the families and relatives of AI patients and to future generations, and more importantly, dentists can work out the best treatment plan for them knowing the exact type of AI they have. After the screening where SMMIP's diagnosis is negative, I'll concentrate my research on finding mutations we have missed in the known genes or to find novel genes with mutations that cause AI. Finding the genetic changes that cause AI helps us learn more about how teeth are made and about all the different ways they can go wrong. With the knowledge and understanding we gain from the tooth genetics, we'll be able to raise the standard of treatment and can hopefully also identify and test new treatment ideas. Finally, a very special thanks to Leeds Doctoral College Research Nights In for giving me the opportunity to share my work with you. If you have any questions or queries, feel free to contact me via my email or Twitter. Thanks for listening.